you can use this as a biomarker and as a potential vaccine or antiviral target, okay? But the function of this protein is not really clear because it's considered non-structural, but it may substitute as a structural protein if it forms a complex with GP2, okay? Some of the pseudotyping data shows that it actually may limit GP cytotoxicity, and as a result, it results in more efficient replication as well as infectivity. Okay, this is a bit of a speculation. So, question then is, how would it do that? How would it make the replication and infectivity more efficient? It's limiting cytotoxicity, so what does that mean? Yeah? So if you kill the cell that you're in, you're not going to be able to use the machinery anymore. So by lowering the cytotoxicity, it's possible that it allows for better replication and infectivity. You can make more particles by making the cell live longer, okay? It acts as a soluble factor as well, which targets the elements of the host defense system, okay? So let's say you have an antibody that recognizes this SGP. What's going to end up happening is you have your SGP that's secreted outside the cell. It's a soluble factor, so it can move around. And if it occupies all the antibodies, or most of the antibodies, are you going to get a good response against the virion? Yes or no? If your antibodies are all occupied with this secreted peptide, what's going to happen? They may not recognize the actual virus as well. So it can contribute to immunosuppression. Question? You have a question? Okay. And also, remember, we are also producing this small 5 kilodalton delta peptide. The function of that is not really known, okay? So what are the possible functions of SGP? You guys can read through the legend, but for example, it can have antigenic subversion. It can act as a decoy antigen, because what it will do is it will start binding to the antibodies because it's secreted outside the cell. It can also lead to inactivation of neutrophils. It can reduce pro-inflammatory cytokines. How would that help? By reducing pro-inflammatory cytokines. How would that help? Yeah. Okay, good. So remember, if it reduces pro-inflammatory cytokines, those cytokines are used to recruit even more immune cells. So you're affecting that process as well. So it's, again, working against the immune system. So this is one of the major questions you should be able to answer. How would this subvert the immune system? Okay? Now, normally, you can restore the function of your endothelial barrier, etc. But as you do that, the virus is really happy to replicate inside these cells, okay? But keep in mind that your SGP can act as a decoy antigen and recruit the cells to itself rather than the rest of the virus, because this is going to be cut and secreted outside of the cell, okay? Everyone clear? Yes? Why is everyone so quiet? What's going on? Exam tomorrow. What exam? Genetics. How many of you have genetics tomorrow? Okay. Any other exams? Maybe? Okay, that's not a good sign. Ooh, right before reading week.
Yep.
That sucks. All right. All right, so continuing on, if you look at a minor nucleocapsid protein, which is VP30, it activates the viral mRNA synthesis in this Ebola virus, okay? So there's a stem loop structure, so again, secondary structure that looks like a hairpin at the beginning of this NP gene that inhibits the RNA polymerase from initiating mRNA synthesis. So this VP30 actually reverses this inhibition, okay? And if you were to disrupt this secondary structure by mutations, what we see is the transcription is not dependent on VP30. So it's mostly for making mRNA. It's used for making mRNA, but VP30 actually does reverse this inhibition of this stem loop structure, okay? It's present in the nucleocapsids, but we do not know the action of this protein. Like, how does it work? We do not know the mechanism of action. So here, for example, you have a leader sequence right here. So why is this 3' and 5'? Because it's negative strand RNA, okay? So you have your leader sequence and the trailer sequence. So in this case, it's the NP mRNA, and there's your secondary structure. So it can be present in the genome itself or in the NP mRNA, okay? Now, if we continue on, what we see is one of the most abundant viral protein that's associated with the envelope is VP40. It's located at the cytoplasmic side of the plasma membrane or the inner side of the viral envelope, okay? So here's your VP4, and if you have, for example, a dimer, it controls how the virus moves through the cell. So it controls the trafficking of the virus, okay? So just like your matrix proteins of other enveloped viruses, it basically allows to form the bridge between glycoproteins to the nucleocapsid. So it basically attaches your glycoprotein to the nucleocapsid. And if you were to express VP40 in mammalian cells, it results in virus-like particles that bud from the plasma membrane, okay? Virus-like particle, not virus. So what does this tell you? Say that again. Okay, so basically it's sufficient by itself for the virus particles to bud, okay? So if you were to just express this protein, you will start budding virus-like particles. They are not actual viruses because there's no genome and there's no other proteins. So that tells us that this protein plays a major role in budding. Now, if you look at the cellular proteins that are involved in, for example, trafficking and sorting of the intracellular vesicles, they can interact with VP40 as well to form the virions at the plasma membrane. So there's a protein called NAD4, which is a ubiquitin ligase, that interacts with it, TSG101, which is a part of the multi-protein complex, which allows to form multivesicular bodies from the endosomes, okay? So these multivesicular bodies are vacuoles that contain many of the small membrane vesicles as well as enzymes. Now let's look at the figures and see how this works. So the VP40 can rearrange to form different kind of functional structures. So dimer, for example, can control trafficking. You can have membrane rearrangement, you can form hexamer for matrix assembly, and it binds to RNA in this way so that it can control transcription, okay? So depending on how many copies of VP40 join together, you might have slightly different functions. And just to show you guys what multivesicular body looks like, you see all these little dots in there? Those are various vesicles that are into this, that have formed this larger structure, which is the multivesicular body. Okay? I don't know how well you can see that here and here. You can see these multivesicular bodies in the cell. And there's one fairly large one, seems like here. Okay? All right. So with me so far? Yes? All right. So let's look at the replication cycle, and you should be able to go over how Ebola virus replicates, okay? And this is, again, this is something that we have already covered, and this is a summary of what's happening. So can you identify all the steps? If you can identify all the steps and all the players involved, you know how Ebola virus works. Question for you guys right now is, how can you tell if this is showing us Ebola virus or Marburg virus? And how can you tell? So remember, Marburg virus is very similar to Ebola virus. They are both filoviruses, and they both cause severe disease. So how can you tell what this life cycle is showing us? Yeah? Okay, so let's assume that those proteins are similar. Then what? There's a very easy way to tell. Yeah? Say that again. Okay, go on. You're on the right track. Okay, but looking at this image, this doesn't talk about RNA editing. How can I tell which virus this is? Just based on this image. Yeah. SGP. Marburg virus does not have SGP. Ebola virus does. Okay, so that's one difference, is major one between these two viruses. Okay. Now, one thing that to add on here is Ebola viruses do tend to form these latent infections. So those who do survive the infection, what we see is that there's periodic spikes in the Ebola virus antibodies even months after their disease has been resolved. Okay? So what that means is there might be latent infection and a potential for disease recurrence. And this disease recurrence can actually trigger outbreaks. So what is recurrence? Basically, reoccurrence of the disease or the symptoms after a period of inactivity. Now, in this paper, the author suggests vaccinating the survivors of Ebola virus, and then that may help prevent disease recurrence, because this will basically boost the protective antibody response in those survivors. But there's more recent reports that also show that Ebola virus persists in the survivors, despite its clearing from blood as well as clinical recovery. So for example, it persists in semen. So as a result, it can have subsequent sexual transmission. If the disease does reoccur, it results in severe organ-specific inflammatory response, okay? And specifically, this could happen at immune-privileged sites, so in the eyes as well as the brain, okay? Now, there was a lethal relapse in the Ebola virus disease survivor, and that survivor then went on and infected multiple people. Okay, so this can happen, and you can study this using non-human primate models. What they showed was that Ebola virus infection was persistent in about 7 out of 36 animals that survived the infection. And this was after antibody treatment. So even after antibody treatment, the disease can persist, okay? And this goes hand in hand with what did we just learn about immune system and Ebola virus. The SGP can subvert the immune system, right? So this could be one of the mechanisms that it does that. And there's potentially a link between 2014 and 2021 outbreaks, okay? Because what they saw was the viral sequences were recognized in the patient even 5 years after the infection. So what that indicates is that somehow these outbreaks are linked, and the Ebola virus genome itself may stay inside our cells. So that's a big question that's still being answered. Okay? Any questions? No questions? Okay, let's continue on. Okay, so we're going to continue with negative strand RNA viruses. And today we are going to be looking at the influenza virus. And just like we have done with all other viruses, we'll look at genome proteins, how it gets in, replication and synthesis, assembly and release. And one thing we're going to introduce today is the antigenic change in the viruses. Okay? So starting with the structure, influenza viruses belong to Orthomyxoviridae family, which is ortho means correct or normal, and myxa is mucus. Okay, so this is based on the ability of the virus to attach to the mucoproteins on the cell surface. So mucoproteins are proteins that are present in mucus. Okay. Now, the myxoviruses themselves, they are split into paramyxo and orthomyxoviruses. They have slightly different structure and replication cycle, but we're going to be focused mostly on orthoviruses, or the influenza virus, sorry. So these are enveloped particles, but they're quasi-spherical, and they could be filamentous as well. So this is an EM image of influenza virus. So it's not perfectly spherical, so it's quasi-spherical, and the envelope is derived from the host membrane by budding, just like other viruses as well. And diameter anywhere from 80 to 120 nanometers, and they have a very compact helical set of nucleocapsids. So again, here's the EM image, here's the model, and here's the schematic. What's the one thing that stands out here in the schematic? Something we haven't seen before yet. Yeah. The genome is fragmented. Okay? So instead of one long genome, you have various fragments of the genome. Okay? So this is, again, RNA virus, negative sense RNA virus that has a fragmented genome. And there are multiple subtypes of the influenza virus, and each type is distinguished by the surface glycoproteins. So hemagglutinin, HA, and neuraminidase, or NA. That's where the naming convention comes from. H1N1, H5N1. Okay, that's how the naming convention comes from. Your hemagglutinin as well as neuraminidase. So there's type A influenza, which infects a wide range of animals, including birds. You have influenza B, which mostly affects humans. Influenza C, again, mostly infects humans and pigs, but it's much more rare. Okay? And lastly, you have influenza D, which infects cattle. And within A, you have various subtypes. And we also have highly pathogenic avian influenza and low pathogenic avian influenza. Avian influenza basically is bird flu, okay? Now, these Orthomyxoviridae, they have two additional genera. One is Thogotovirus, which is transmitted by ticks, and Isavirus, which infects fish, mainly salmon, okay? So as you can imagine, these viruses, they do have a strong economical impact, especially when it comes to livestock or even making us sick, okay? How many of you had your flu shots yet? Okay, 2 people. Get your flu shots, guys. It'll help you. All right, so if you look at influenza A viruses, as you can see, it can have a variety of hosts that it can infect. So for example, again, we have bats, we have aquatic wild birds, domestic poultry, marine species, pigs, other animals, and obviously humans, okay? So again, what kind of spread is this? What do we call that? Zoonosis, zoonotic spread. So it jumped from animals to humans, for example, or they jumped from one species to another. So first thing, let's look at the disease and spread. Symptoms are high fever, sore throat, cough, headache, and muscular pain. Almost everyone here probably has had flu, yes, at some point in their life. It's not pleasant, okay? But it's fatal in elderly, infants, and people who are chronically ill. And this usually happens by secondary infections in those patients. So for example, influenza A virus, it causes serious acute disease in humans. You get sporadic pandemics and annual epidemics every year. So that's why we have to get a flu shot every year, okay? So as a result, this is one of the best studied viruses, okay? And just to give you guys a number, there are about 20,000 deaths per year just in North America because of these flu viruses. And there are emerging avian influenza virus strains, and they continually threaten birds, and they have a potential for another human pandemic. So please make sure you get your flu shot. Another question. The flu cases actually dropped quite a bit during COVID-19 pandemic. Why do you guys think that's the case? Let someone else from this side give a chance. Who wants to answer? Okay, people were quarantined. When you went out, you were wearing masks. You were using sanitizer, washing hands. Okay? So all that does help. So what that means is you should be continuing those practices of washing your hands, sanitizing, and if necessary, wearing a mask. Okay? But the problem here is the symptoms are common to a lot of other infections as well. So for example, if you just have a minor cold, you have flu or COVID-19 or RSV, you get similar symptoms for all these conditions. So you'll have to really evaluate and test to see what might be actually infecting, okay? COVID-19 is easier. We have home tests that we can use like we did before. RSV, cold will be, again, or flu will be a little bit more difficult to diagnose, okay? So you have to be very careful, and usually doctors are able to figure out which one is which, okay? They might order some tests if necessary. All right, so the influenza virus, it causes infection of the respiratory system, as you guys already know. and it can lead to secondary bacterial infections. So for example, pneumonia, by providing easier access to the lung cells. Because what ends up happening is the virus infects and causes a loss of the epithelial cells that line our lung. But one feature they have is that they are ciliated. Why are they ciliated? Because they move mucus. These cilia, they move mucus, which is called a mucociliary flow. And if you disrupt this, what's going to happen? Yeah. Okay. So it will be hard for you to clear your respiratory tract, and as a result, you are more liable to infection. So the flow of mucus and debris along the epithelial lining stops or reduces, and as a result, you can have secondary bacterial infections. Because remember, mucus acts as a protective layer. A lot of the stuff gets caught in the mucus every single day, and it gets removed from our bodies, okay, or it's destroyed. So if you disrupt this whole process, all the bacteria, for example, that were stuck in that mucus, they're staying there in your lungs. And as a result, they can start replicating, start dividing, and cause a secondary infection. So virus also induces the production of interferons and cytokines, and as a result, you get a local and systemic inflammatory response. So here's a TEM image of the cilia here, and here's the SEM image. So you can see it's a fairly thick coat, almost looks like grass, okay? And these cilia beat to remove the mucus, but if you damage this system, it's not going to work as efficiently, and as a result, cause secondary infections. Now, if you look at the genome of this, it's a segmented, negative sense, single-stranded RNA genome. It can have anywhere from 6 to 8 different segments, which are basically in the conformation of a helical nucleocapsid. So the viral particle will have multiple helical nucleocapsids. A single virion will have a complete set of the genome fragments, and the total genome size can be anywhere from 10 to 15 kilobases. Now, a couple of things I want you to pay attention to here. This Uni12 and Uni13 region, okay? That's the universal primer region, okay? These are self-complementary. So what do you think will happen? What does it mean that they are self-complementary? Is the sequence of this region complementary to this region? Yes or no? Look at the sequence. Is it complementary? Yes? Have a closer look. Is it complementary? Sorry? Hard to tell? Okay. So there's some complementarity between these regions, and basically what it does is it helps fold the genome like this. Okay? Instead of having this long segment, you make the RNPs like this. Okay? And you have your non-coding region on both ends. You have packaging signal as well as the coding region. And each of these segments will have this kind of architecture. You have your uni regions, you have your non-coding regions, and you have a protein coding region. So in this case, each segment is encoding a different protein. So PB1, 2, PA, HA, NA, and so on. Okay? Everyone clear? Yes or no? Okay. And this is all the different fragments, what the proteins they encode, the size, and the function of the protein. So you can look at the summary in the table. And we're going to go through some of this in detail in the coming slides. Okay, let's take 10 minutes, and then we'll continue. Okay, before I forget, looks like this lecture 7, only half of the recording was there. Yes? So I've contacted the support. Hopefully they can find the file. And once they do, I'll post the original file. Yeah. That's what I'm saying. So it only recorded 30 minutes, right?
No, it doesn't have any sound.
It doesn't have what?
Any sound.
It doesn't have any sound.
There's only the picture over there, but there's I cannot hear it talking.
Okay, that's weird.
So it's 30
Okay.
minutes long and there's no audio.
Okay, I will send them another email, and hopefully they can find the file. Okay? Yeah. The quiz is on lecture 6. Or did we have quiz on lecture 6 already or no? So lecture 7. Are we on lecture 6 or lecture 7 for the quiz? I can't remember. Sorry? Okay, quiz 5 I know we finished, but was it on lecture 6 or lecture 5? It's based on lecture 5, so quiz 6 would be based on lecture 6. Okay? So hopefully we'll solve this issue before your next quiz for lecture 7 comes up. All right, 10 minutes, then we continue.
we're looking at mainly? Like which genre is it?
So influenza viruses, like,
It just covers like
it
the whole
covers
family.
the whole family. Yes.
Whole family.
Yeah.
And another thing is I noticed in this image there's RNA polymerase.
Yeah, we'll go over that.
Go
So
over that?
because they do package
For
sometimes.
each of them. Okay.
Yeah.
And then this one has an ion channel as well.
Yeah,
Okay.
the M2 ion channel.
Okay.
It's coming.
Okay. I was just curious because
Yeah.
I just
So M2 channel is what allows for protonation, like movement of protons.
yeah.
So there's a drug that actually inhibits